Have you ever wondered how fingerprints on evidence are made visible in crime labs? Or what makes this process possible? If you're interested to know, keep watching the video. Hi everyone, my name is Zeina Chamseddine, and today I'll be talking about a chemical reaction that brings hidden clues to light, which is the Ninhydrin test. In 1910, a surprising breakthrough happened in Cambridge lab. While studying the shifting forms of cyclic ketones, a chemist named Siegfried Roman discovered something unexpected, the Ninhydrin reaction. This accidental discovery would go on to change how we understand amino acids, peptides, and proteins, and eventually become a powerful tool for uncovering hidden clues in forensic sciences. So, what is this mysterious chemical test we're talking about? Let's take a closer look. Imagine this. Every time you touch something, tiny traces of amino acids from your sweat are left behind, invisible to the naked eye. But with a little bit of science magic, specifically the Ninhydrin test, those invisible traces can reveal themselves to vivid color, helping scientists identify fingerprints in forensic investigations. Of course, the purpose of this test goes beyond just forensic science. It's actually a powerful tool used by scientists to detect the presence of amino acids, peptides, and proteins. In this slide, you can see the illustration of the protein structure. You can clearly see how amino acids are linked together in a complex chain. To prove the presence of amino acids, we use this chemical, which is the Ninhydrin compound. This compound is a solid compound that's typically found as a white to pale yellow crystalline powder. Now it's time to dive deeper into the actual chemistry behind the Ninhydrin reaction and see how it reveals those hidden amino acids. So when the Ninhydrin comes into contact with the amino acid, it specifically reacts with the amino group of that amino acid. This reaction leads to a fascinating color change that's central to its applications. On the left here, you can see the general formula, where the amino acid reacts with the Ninhydrin in order to produce the Roman's complex, or the Roman's purple, which is a complex that has a maximum absorption wavelength of 570 nanometers, which is responsible for its purple color. And on the right here, you can see the proposed mechanism for the reaction between Ninhydrin and the amino acid, and how it produces the Roman's purple. Now, as you can see, this complex is independent of the R group. This is because the R group has departed. And this also explains how almost all amino acids, in spite of their different R groups, react in the same way with the Ninhydrin to produce the same product, which is the Roman's purple. For the procedure, first of all, we start by preparing our Ninhydrin solution by dissolving 0.2 grams of Ninhydrin in 50 ml water. Then we take 1 milliliter of our Ninhydrin solution and we add to it 2 milligrams of our desired sample. Then we heat this sample in a water bath. A positive test is a purple test, which indicates the presence of amino acids. If there is no color change, we get a negative test, indicating the absence of amino acids. And when a yellow color is present, this indicates specifically the existence of either proline or hydroxyproline and other certain aminobenzoic acids. And these compounds react differently with Ninhydrin compared to other amino acids. Now it's time to discuss the importance of the Ninhydrin test. This test is a critical tool in chemistry and biochemistry because it helps identify and measure the amino acids and proteins. I just said measure, right? And I previously said it is a qualitative test and not a quantitative test. It is true that this test is qualitative, as I said before, but it's important to highlight that when using an automatic amino acid analyzer, this device detects the intensity of the color produced and translates it into quantitative data, allowing scientists to measure the exact amount of the amino acids in a sample. For the limitations, some proteins might have blocked or modified amino groups, which prevent them from showing a positive result. This can lead to false negatives, which is important when interpreting the results in research or diagnostics. Also, proline is unique among amino acids because of its secondary amine group. Since it has a slightly different structure, it gives a yellow color instead of purple, making it stand out in the test results. These are the references that I used. Thank you so much for watching the video. Let's keep our energy and curiosity high for exploring more and more in the magic of science.